So, how do cells get energy out of food? The process called cellular respiration. So basically, we're talking about the release of energy from glucose to make ATP, because we said that is the energy coin that the cell uses. This is an aerobic process, meaning that it needs oxygen. That makes sense. You cannot, I know cellular respiration is not the same thing than breathing, but you breathe to take in oxygen for cellular respiration. Without oxygen, you don't live very long, do you? And it produces carbon dioxide. Carbon dioxide actually, and water. I don't know why it's not mentioned in there. So that's the reaction. This is glucose, react with 6 oxygens and produces 6 carbon dioxide, 6 water, and 32 ATPs. And we are going to discuss this number a little bit later, okay? Why 32? But yes, you need to know this reaction. Okay? Now, the good thing is that we're going to be looking in the next video to photosynthesis, and you're going to see it's basically the same reaction backwards. So you memorize it once, you can use it twice. I need to introduce 2 molecules because you have a pathway of cellular respiration that breaks down glucose, but to be released slowly. That is key. You know, if you could burn glucose, you can do that. I know it's not glucose, it's dextrose, but you can get sugar, and if you heat it enough, it catches on fire. Okay, 32 ATPs is a lot of energy. If they all 32 were released at the same time, it would burn the cell to a crisp. It would kill the cell. So the idea of cellular respiration is not only to release energy, but to release it slowly where the cell can handle it. Okay, so it involves many steps. During those steps, we are going to see their oxidation steps, so they're going to release electrons. So you need electron carriers. Okay, the 2 electron carriers are NAD plus and FAD. Each one carries 2 electrons and 2 hydrogen atoms, because let's face it, I mean, if you have hydrogen, you break it down into a hydrogen ion and an electron, so they tend to go together. But what we're interested is in those electrons, because they're high energy electrons. Okay, they're going to carry those electrons to the electron transport chains, where the bulk of the ATP is going to be formed. So cellular respiration is made up of 4 different phases. I'm using this particular textbook. Yours may say only 3 phases, because some people consider prep reaction part of the citric acid cycle. Some people use different terms, okay? But we have glycolysis that takes place in the cytoplasm. The preparatory reaction, I like this book where it considers a separate one, because it's moving your reaction from the cytoplasma into the mitochondria. It's trying to cross into the mitochondria. The citric acid cycle takes place in the mitochondrial matrix, and then you have the electron transport chain and oxidative phosphorylation that takes place in the inner mitochondrial membrane. Remember, the mitochondria have 2 membranes, or we call those cristae. Like I said, some books will consider the prep reaction and the citric acid cycle part of the same, because they take place in different locations and they have different purposes. I agree with 4. But obviously you want to say whatever your book says when you're taking an exam. So the first step is glycolysis, and we said this takes place in the cytoplasm. Something that's important, because we'll come back later on, it does not require any oxygen. Now you're going to say, what kind of formulas are those? No, these are not the real formulas. I'm just showing you the carbons, because I don't want you to memorize formulas. Believe me, this is not biochemistry. If this was a biochemistry class, you will study every individual reaction and what are the reactants, what are the products, every detail. This is kind of like overall picture of glycolysis. You get a glucose molecule and becomes 2-pyruvate. So you have a glucose molecule and eventually you have 2-pyruvates. Don't worry about the intermediates, okay? At a certain point, the 6 carbon, you have 6 carbons in there, breaks into 2-3 carbon molecules that continue. But notice these reactions, some are going to use ATP. At the beginning, you have to use 2 ATPs. Okay? You're going to be producing electrons that are going to be picked up by NAD+, so it produces NADH, 2 of them, and produces 4 ATPs because you produce 2 in each side. The net gain, if you form 4 ATPs but you use 2, is going to be 2 ATPs in glycolysis. Okay? So 2 ATPs, 2 NADHs. Like I said, don't worry about the reactions in between or anything like that. If you want to know the details, you can take biochemistry. Now the problem is pyruvic acid cannot enter the mitochondria. So pyruvic acid in the prep reaction, it's turned into acetyl-CoA, a molecule that can cross the mitochondrial membrane. Okay? Now it happens twice. Why? Because every glucose produces 2 pyruvic acids, but each one is going to react with the coenzyme A, releases carbon dioxide, releases electrons, and makes acetyl-CoA that goes into the next step, the citric acid cycle. All you're doing is taking the pyruvic acid that was in the cytoplasm and takes it into the matrix of the mitochondria where the Krebs or the citric acid cycle can take place. So you have 2 pyruvic acids, you need 2 acyl groups attached, you have 2 acetyl-CoAs, all of this times 2, so you produce 2 carbon dioxides, and you form 2 NADHs. We are going to get back to all of these NADHs because these electrons are where most of the energy resides, so obviously we are going to have to use them. The citric acid cycle, depending on your book, I don't think there's any set of reactions in biochemistry that gets more name. Some people call it Krebs cycle after the person who discovered the cycle and isolate all of the different elements. We're not going to go through all the reactions, but describe all of the reactions and all that. So it makes sense to name it after him. Citric acid cycle is named because the first reactant form is citric acid. Some people call it I hate when it does that. So apologize. My pen sometimes does things like that. But some people call it tricarboxylic. Acid cycle because there's 3 carboxylic acids in the cycle. And when I was a graduate student, we just call it TCA for tricarboxylic acid, TCA cycle. Different books use different terms, and there's not the preferred one. It's just a matter of opinion. So the book I'm using here uses citric acid cycle, but keep in mind it may have a different name in your book. So the acetyl-CoA that we had from the prep reaction, remember we had 2 of them. I don't know what's wrong with my computer today. But citric acid cycle turns twice because you put in 2 acetyl-CoAs. Okay? Notice that every turn, what you're putting there is a 2 carbon piece because we had lost 1 before, so you're going to form 2 carbon dioxides. But you're going to release a bunch of electrons at NADH or FADH2. And you're going to also form 1 lonely ATP. But this is 1 turn. Remember, all of this is going to be multiplied times 2 for 1 glucose molecule. So we form a little bit of ATP in glycolysis, we form a little bit of ATP in the citric acid cycle, most of the energy is stored in the NADH and FADH2 that are the electron carriers that have picked up those high energy electrons. So they pass the electrons to the electron transport chain. Very important before I say anything else, the final electron acceptor is going to be oxygen. This is one of my favorite questions to ask in an exam. Okay? This is very important because basically you have oxygen that picks up the electrons. There's obviously prons everywhere in the cell, so it produces word. Remember, that was one of the products of cellular respiration, word. This is where the word comes from. The reason this is important is because we say without oxygen, there's no cellular respiration. That's the reason why. Rat poison, you know how that works? It prevents that final transfer of electrons to the oxygen, so the rat cannot produce energy, it dies. Okay? What we have in the electron transport chain is a set of molecules, and we're not going to get into details, that supports a series of oxidation reduction reactions. That means we are passing electrons along. It's like a bucket brigade. A molecule picks up an electron and passes it on to the next molecule. And then that molecule passes the electrons to the next molecule. But there's a few places where when you transfer the electrons from one molecule to another, enough energy has been released to pump prons, but we're going to get to that in a moment. For the time being, it's just a bucket brigade passing on the electrons, and they're located into the inner mitochondrial membrane. Okay? But basically, at different spots, they are going to pump protons into the space between the two membranes, so we call that the intermembrane space. What does have to be pumped? What does it take energy? Because there's a lot of protons already in there, and you're adding more. It's active transport. Okay? The one thing I want you to notice is that NADH transfers the electron to the very beginning. So there's going to be 1, 2, 3 locations where you can pump protons. FADH2 transfers a little bit later down the chain, so there's only 2 locations where they can pump protons. And the protons that have been pumped, they are the ones that are going to eventually produce ATP. So we see that NADH and FADH2 produce different amounts of energy. But for the time being, we are pumping protons from the matrix into the intermembrane space. They accumulate there, and they create a concentration gradient. Now, protons cannot go through the plasma membrane, okay? So they accumulate in there. It's like a dam that's holding water. Now, the inner membrane contains an enzyme that's called ATP synthase. That's actually both an enzyme and a channel. It allows protons to flow. Obviously, they're going to flow back into the matrix because there's a high concentration in the intermembrane space, there's a low concentration in the matrix, so they're going to flow from high to low concentration. But as they go through it, this is like a turbine, except that instead of producing electricity, it produces ATP. Okay? This process is called chemiosmosis and accounts for 9% of the ATP produced in aerobic respiration. When we get to evolution, I'm going to spend some time about ATP synthase because it's a real turbine. It's very small. You can imagine it's in the membrane of the mitochondria. That's a teeny organelle inside a very small cell, but it's made up of many different parts. Like in a turbine, when the protons flow, you have parts that rotate and move. And like I said, instead of electricity produces ATP, but it's a marvel of engineering. We are not anywhere close to be able to make something like that size. Okay? So how much ATP do you get? Because at the beginning we said 32. Actually, the number varies. Varies depending on the cell. Okay? Liver cells produce more ATP than most other cells. Why? Because remember, during glycolysis, we form NADH, and that stays in the cytoplasm. But in order to be used, it needs to be carried inside the mitochondria. NADH cannot cross the mitochondrial membrane. So different cells use different methods. Sometimes the electron is donated to an NAD inside the mitochondria. Sometimes it's donated to an FAD inside the mitochondria. There are carriers that carry the electron from the outside to the inside, but who do they donate the electron to varies. And we know NADH produces more ATP than FAD. So that's the reason the number is variable in there. 32 is considered kind of like the standard, the average number of ATPs. Some people say, we just use, say, 4% of the energy in glucose. That's not very efficient. Actually, if you compare to your car, it's unbelievably efficient. Now, we said oxygen is necessary. So what happens? You know the cells that live whenever there's no oxygen in there. So whenever there's no oxygen, this is called anaerobic conditions, no oxygen. How do the cells produce energy? And the answer is fermentation. Okay, let me make it clear. Fermentation does not produce ATP. Let me just add that, because again, this is one of my favorite questions. Does not produce. So what is the point of fermentation? Well, you could be doing, actually, let me change this. In glycolysis, you do produce a bit of ATP, and eventually the NAD is regenerated. Remember, NAD plus picks up the electron, becomes NADH plus prons, and then when it gets to the electron transport chains, you regenerate NAD plus. Right? So everything is fine. But what happens if there's no cellular respiration? There's no electron transport chain because there's no oxygen. Glycolysis is going to stop because very soon it's going to run out of NAD plus. When you have used up all of the NAD plus, that's it. So the idea of fermentation is to regenerate the NAD plus, because if you regenerate the NAD plus in fermentation, now glycolysis can continue, and we say glycolysis produces 2 ATPs, which is not a whole lot compared to 32, but it beats nothing. Okay? So that's, again, one of my favorite questions. What is the purpose of fermentation? Fermentation does not produce ATP. Fermentation regenerates NAD+, so fermentation allows glycolysis to continue. And there's two main types of fermentation, depending on the product. So we have lactic acid and alcohol fermentation. You find lactic acid fermentation in red blood cells, in the muscle, whenever there's not enough oxygen supply. I remember that my son was running long distance when he was in high school and in college, and if they were not in good shape, they went anaerobic very quickly. The heart could not pump blood fast enough, so the muscle went anaerobic, and they couldn't work properly. Okay, they switched to fermentation. Lactic acid accumulated, and lactic acid causes a bunch of problems. We're not going to get into that. This is not anatomy and physiology. Okay, but at least the muscle was able to do fermentation. Glucose was broken into pyruvic. Glycolysis takes place as usual, but the NADH was regenerated when the lactic acid was formed. So you get NADH, and glycolysis continues, and you get some energy, not a whole lot. Lactic acid must be removed from the blood because of the problems that it causes. But in case of when you're running anaerobic, just take whatever you can. Okay? Alcohol fermentation is more common in yeast because in this particular case, the pyruvic acid undergoes a series of reactions that produce ethanol and regenerates the NAD+. So once again, you regenerate the NAD+, for glycolysis to continue. But the products are alcohol. That's how you make beer and wine and all of that. The carbon dioxide is used in baking. Whenever you want to raise that, you're making bread. I had once a student, and at this point, she screamed and left the room. We all look at each other. I don't know. She came back later on. She apologized, and she told me that she was very, very careful when she fed her family, and she baked her own bread because she did not like them eating preservatives. And it's difficult to find bread without preservatives, and she didn't realize she was feeding them alcohol. And I told her, don't worry. When you put the dog in the oven, the alcohol evaporates. There's no alcohol in the bread. Really? She was so happy. You see, if instead of running out of the room, you ask questions, I could have saved you a big upsetness. So carbon dioxide, like I said, is used to raise the dog. It's used in some alcoholic beverages to add gas to it. You know, like if you drink champagne, that's what we talk about having carbonated alcoholic drinks. But what most people use it for is to produce ethanol. Now, what happens to the other food? Because this is very easy. If you have glucose, I see how you get energy. But I don't need glucose. I eat steak and potatoes and rice and vegetables and all of that. Okay. Carbohydrates can be broken down into glucose. Okay. Some amino acids, and we'll cover all of this mostly in the second semester, and glycerol, they all go into glycolysis. Carbohydrates, starch, no cellular, but oh my goodness, the animal carbohydrate, whatever, they produce glucose that goes into glycolysis. Fatty acids and the other amino acids, they're going to form an intermediate that can go into the pyruvate oxidation or the intermediate reaction. Okay? Other amino acids break down into two carbon pieces, so they go directly into the citric acid cycle. They all end up at the end in the oxidative phosphorylation producing energy. They just have to be transformed into different intermediates of cellular respiration. Yes, this is starting to get complicated, and I do have a beautiful worksheet, actually a table for you to fill in, where you look at each stage of cellular respiration, what happens in that stage, where in the cell it takes place, what are the products, reactants, is there anything special, and it helps you summarize this whole chapter into an easy-to-see chart. I'd be glad to share with you if you make an appointment with me. Again, the link to my wife's account is in the video description. I'm not just trying to sell it, but that particular worksheet will include photosynthesis too. So not only do you summarize two chapters in one table, but you can see the similarities. And when you grasp the similarities, it makes memorization a whole lot easier. Okay? So click on the link, make an appointment, and let's work together.